the PI of the P60 Multidisciplinary Clinical Research Center, which is co-sponsoring this event. So I'd like very much to thank the people who did all the work of putting this together. The Shan-Ching Kui, who you'll hear from, and Jake Chen have been instrumental in that, and Dr. Cimino has been wonderful to work with on this. Without the transformational hiring of Dr. Cimino and Dr. Chen and others, we would not be able to talk meaningfully at UAB about bioinformatics and collaborations that we have. So we're very pleased to have them here with us. I'd also like to thank Stephanie Ledbetter, Carol Ballinger, Paula Kiley, and all the others who helped put this together. So thank you all very much for coming. I think it'll be a very fruitful day, and please enjoy. So our first introductory speaker is Dr. Cimino.
Thank you, Lou.
Thank you.
Okay, wow, great turnout. Is this oh, it's just recording, okay. Can you hear me in the back there? Okay. So thanks for this invitation. They wanted somebody from the Informatics Institute to open the session who wouldn't actually be presenting at the session. And since I'm the only one that doesn't know anything about bioinformatics, that role fell to me. So we'll tell you about where things stand in the Informatics Institute and in relation to bioinformatics and translational informatics. And hopefully that will set the stage for the rest of the talks today. So I had this really nice diagram of various levels of biology and medicine and informatics. It starts off with the bioinformatics, which is down at the level of biology of molecular biology, cellular biology. We move up to the cell and to organisms, and we get into translational informatics, and we move up to the human. Then we start talking about pathophysiology and physiology and clinical research informatics. And then finally we get to the level of the patient, which actually is why we're all here, believe it or not. You may think you're working on zebrafish, but you're all really actually interested in not in the health of zebrafish or fruit flies, but health of humans. And so that's where patient care comes in and clinical informatics, and then we can even extend that out to the population and to public health informatics. So the topic of this conference is down at that end of the spectrum, the translational bioinformatics, which is a nice term to kind of bring together two areas very closely related and often very difficult to separate out. What do we mean by bio? What do we mean by translational? And we solve that problem with the joint phrase. Okay, so I look at informatics at the institution. I've been here 2 years, and the Informatics Institute started with me, but it really didn't officially start till we got somebody else here besides me, and that was Jake Chen, who came just over a year ago, and we've been building faculty since then, and that has added to the informatics capabilities at UAB, which was the intention of creating the Informatics Institute in the first place. So we can look at bioinformatics support, and there's been a very strong a tradition of that here over the last, I don't know, decade probably, of providing support for researchers who need help in analyzing their biological data. And then we also have clinical informatics support that's been a bit more recent, maybe over the last 5-6 years, with the introduction of the Clinical Data Warehouse and I2B2 to provide access to patient data that can be used in research. And those have been really subsumed by the Center for Clinical and Translational Science, chaired by, the PI is directed by Bob Kimberly. So the CCTS and clinical informatics, that was kind of it. If you wanted informatics help, that's where you went. That was sort of the central focus for UAB. Then we had the Informatics Institute come along, and now my charge has been to enhance that, not simply by adding to that work, the work that's done there, but bring in new aspects to that. And I've been doing that through bringing in informatics faculty who provide a new dimension to support for translational bioinformatics research. So there's my bioinformatics faculty that have been added. Upper left there is Jake Chen, and the others have all been added. Some are speaking today, have been added to the pool to work with the analysts and informaticians in CCTS to enhance the bioinformatics capability. We also have clinical informatics support. I'm a clinical informatician, and we have a couple others who are going to work in the clinical end of things, but not the topic for today. So what are they going to do? Well, you know, we talk about, when we're talking about tenure at least, we talk about things like education, research, and service. And there's this kind of funny area that's in between research and service that has been called facilitation of scholarship. And what that means is you're kind of doing scholarship, but you're really helping somebody else with their scholarship. So you help them solve their problem, you bring your expertise to the task. It's enough of an intellectual contribution that it's not an acknowledgement, it's a full co-authorship, a co-PI or a co-investigator on a grant, but it's not quite at the level of your own research, which may be, in the case of informatics, developing new informatics methods and approaches that can then be applied to other problems. So, oops, I gave my, what happened? Oh boy. So you would think I know something about computers. Somebody hit, let's see if we can get this going again. That is our scholarship. Thanks, sorry about that. Our scholarship is the development of new informatics approaches and methods that can be applied to the problems that other people are developing or trying to solve. Ideally, what we would do is build things that you would then use in your research. In practical matters, we have two other things that we have to do. First, we work with you to solve your problems, and in the process, we gain insights into more generalized solutions. And then we have to develop those solutions into some kind of tools, pipelines, what have you, that could then be applied in your research. So an example of that in the clinical side is I2B2. I2B2 is a clinical data warehouse. Originally, informaticians like myself work with researchers to figure out what kind of data do people need from electronic health records, how do we construct the queries, how do we pull the data and give it to them, and eventually from that work, clinical informaticians at Harvard developed I2B2, which is a self-service tool that developed by informaticians for use by clinical and translational researchers. So that is the scholarship of facilitation that we're trying to bring with the Informatics Institute. And you're going to hear from a number of members of the Institute today, and I think you'll get a sense of what kind of a different level, a meta level, if you will, of scholarship. Okay, and where does that all fit in? Well, it all comes down to really precision medicine. So that is the new hot topic, or you may hear it called personalized medicine. We have a new Personalized Medicine Institute here as well. And so precision medicine is the place where we're going to bring all this together, both the bioinformatics, translational informatics, clinical research informatics, and clinical informatics, to bring to bear to the actual improvement and the care of patients, starting at the molecular level and working all the way up to the organism. So I'm happy to take questions, although I would expect that you don't have any from me, and it's really the other speakers that you're going to have questions for. Yeah. I don't know if we're going to get a microphone to you in time here, but
that's okay. So mine is a general question with respect to the CCTS and this facilitation of scholarship or scholarship of facilitation.
yeah, it's just recording.
Oh, it's just recording. Okay. So is there any efforts to provide perhaps a demonstration of some examples to give investigators ideas of what might be provided through the CCTS to provide more fodder for a consultation rather than just going in from scratch with perhaps a very general or vague idea of what might be needed
Yes.
to kind of guide those conversations.
So the question was, does the CCTS provide any kind of examples or guidance for people who are trying to figure out how to get take advantage of these kinds of techniques and resources? The answer is certainly yes. There's a CCTS newsletter that comes out every week, every Friday, that lists all of the events that are related to that. We have one this afternoon at 4:45, I guess, in the PCAMS building, and we have them every month. We have those forums every month, but there are many other events going on, and this is one of them. But there's also there's workshops. There's a gateway meeting, which I'm blanking on the exact time and date, where people can actually come and bring their examples. Sorry, was it?
Second and fourth Wednesday.
Second and fourth. second and fourth Wednesday, 2 to 3, also in PCAMs, where people bring their examples, and that's open to the community if you want to come to that and see how other people are making use of informatics resources. But I would recommend going to the CCTS website, sign up for the newsletter if you don't already get it, and read the newsletter because it's got all those events in it. Okay, so with that, I'm going to turn it over to our next speaker. Is it Jake? Who's next?
Well, you are early.
I'm early, yes. Well, listen, leave me here. I'll break something else. There you go.
So, Alex, I don't need it. So I'm the chair. So
That's fine. If you have anything you need to say, though. Yeah.
Alex, do you want to load your thing? Sure. Yeah. So we're going to start our first session. So this afternoon, if you look at your agenda, it's organized into 2 sessions. After each, there are 4 speakers in each session. And then you will see there is a 30-minute discussion, panel discussion, and you can ask questions, or and the speakers will be all standing there. You're welcome to ask each one after their presentation, just in case we run out of time, and you can ask them during the panel discussion. Let's see, Alex, are you loaded? No, right?
It didn't show up yet.
And so our first speaker is Dr. Rosenberg, and so he is a fairly new member, right? For Ron? Yeah, a new member of the Informatics Institute and the Bioinformatics side. And he has a lot of experience in both academia and industry in terms of software development. And today he's going to talk about his major effort in visualization of iRepertoire, right? So if you have a new system like Repertoire data and how to look at them and how to analyze them, this is the guy to come to. And he's the associate professor in the microbiology department. Is there a good summary of what you do?
Give me just a second. This computer.
Yeah, we have a slight technical thing. So if you guys, any of you have questions, it's a good time to bring it up for Lo or for Jean or even any of the speakers here you see. Or you can focus on eating your lunch if you haven't finished it yet.
What's the name of it? The only thing on the
And when we talk about informatics, and technical glitches are not rare things. So give a little taste today. When you try to do something by informatics, be patient, and there will be glitches. And we are early, so. Okay, so the second one? Yeah.
Yes, you do. There you go. Just stick that in your pocket.
I think this is the pointer.
It's actually, this is actually, I don't know where this one came from.
Oh, okay.
This is it's just the forward and backwards. Nope,
Okay.
just the forward,
Okay.
back, just those 2 buttons.
Okay.
That's it.
That one has a pointer though. I might use
Oh,
the
okay.
top one. So
Well, let's do this then.
Oh. Okay.
We'll switch them up.
Anyway, thanks everybody for being here today, and also thank you for the organizers for putting this together. I think this will be a really useful symposium, and I'm anxious to hear about what my colleagues are going to do because I'm fairly new here, as mentioned. And so today I'll be talking about two projects that I'm going to highlight. And these are projects that I did while I was at the University of Rochester. And one of them, the repertoire sequencing, is one that I'm still, thank you.
Got a laser.
Okay.
That's the laser, that's the front and the back.
Great.
The other one wasn't responding, so just.
Okay. All right, so I'll be talking about two areas, basically that both very useful in immunology research. The first one is a fairly new application of next-gen sequencing for understanding B-cell repertoires. And the second topic I'll go over briefly is a new approach to a course in immunology research, which is multi-parameter flow cytometry. I'm not going to talk explicitly about data visualization, but I'm hoping it'll come through with a discussion of the other two projects, that this has always been a fairly big thread in my work. So briefly, just want to introduce myself to the community. I was at the University of Rochester for the past 7 years in the Department of Medicine, Division of Allergy, Immunology, and Rheumatology. And prior to that, I worked in industry. I started out my career in Monsanto. That's my first microarray from a long time ago. That was an in-house produced cDNA array on nylon membranes. And at the time was considered a big data set. It was 6,000 elements. Then I worked at a couple of startups and then did some various consulting in a variety of different platforms. So the first topic will be B-cell repertoire sequencing. And I'm not going to read this slide, but I just want to point out it's a fairly new application where we sequence the immunoglobulin gene of sorted B cells. And it has a lot of applications in immunology, so we could answer questions about lineages and relationships between subtypes of cells. We could use it for diagnostics or diagnostic applications in cancer or potentially in autoimmunity. It's very useful for vaccine research, understanding dynamics of immune responses, and also it can be used in conjunction with single cell for monoclonal antibody generation. And although we haven't used this platform for T-cell repertoires, it's certainly doable to apply the analytics that we've built for T-cells as well. So you can answer questions about related to tumor sequencing, where you can sequence both tumors in peripheral blood or different tissues and look for lineages that are related between them. So just to get everybody on the same page, this is the gene that we're sequencing. It's the immunoglobulin gene. It has 3 components that is transcribed to make the antibody. And it has a V, D, and a J region. And each of these are the result of recombination during B-cell maturation. So there are different V segments, D segments, and J segments that are available. And these recombine to introduce diversity into the naive B-cell population. And that naive B-cell population can then, during an immune response, be used to generate even more diverse repertoire through mutation and during an immune response. And that mutation mostly occurs in the so-called CDR regions, and especially CDR3, which make the antibody receptor. And the way we've been using this for B-cell biology is to sort cells. So a lot of investigators do this with bulk PBMCs or B cells. We actually sort the cells into different populations. And so this is a diagram showing B-cell maturation in lineages. So we typically start here at the naive B cells that have a diverse repertoire, meaning that they have the potential to encode lots of different antibodies. And then during an immune response, there's proliferation, introduction of accumulated mutations, mainly in the CDR regions, that's represented by these circles, and they can happen in different directions. And so from this, you can get a lineage tree, like so. And so what we're doing in this pipeline is to sequence the immunoglobulin genes from a sample, and then trying to reconstruct what these lineages are, and visualize it and apply different analysis techniques to it. This is an overview of the analytical environment that we've developed. So the wet lab component is up here. We have samples, we sort them using facts, we generate amplicons and then that comprises the library, and we can do this for different cell subsets. We run them through a MiSeq. We have a 2x300 reads, and this is paired-end sequencing. So each sample generates 2 data files as the output. So 2 FASTQ files for a forward and reverse read. And then each one of these pairs goes through a pipeline that consists of both off-the-shelf and in-house developed components. And it does things like quality filtering, joining the sequences, quality filtering, determining the isotype. We send these sequences out for alignment using vQuest, which is something that's not directly integrated into the platform. We have to actually send it to a website, but we can do this at 500,000 sequences at a time. And then we can take different samples, the data from different samples that are aligned, and we can combine them in this lineage assigner. So essentially what we're doing is we have sequences back that are quality controlled sequences that are annotated as to which VD and J segment they come from. We have mutational statistics. Then they're grouped together based on these lineages, based on a set of rules that we can define. So for example, we can say a lineage is when 2 sequences belong to the same lineage when they share the same V and J segment. The CER3 length is the same. The junction regions are the same. And the CER3s all have a certain homology with each other. And that's an example of a set of rules that can be used to define a lineage in this context. It's not exact, and it's not based on actually measuring the real lineage. It's an approximation. It's a clustering method to organize the sequences together. Then all of this data gets dumped into a single container from which all of our analysis tools can operate. So the original incarnation of this platform generated thousands or tens of thousands of files, and it was just too unwieldy to work with. So each container is an SQLite database. And so if you are familiar with SQL, you can interrogate it directly through a terminal, and you can ask very specific questions. So for example, you can say, compare what is the average CDR3 length for sequences of plasmablasts from this particular sample that have this particular motif in their FR1 region compared to those that don't have that motif. And those are questions that are really easily answered directly through SQL. And you can imagine with this kind of data, there's a really broad range of questions you can ask. But for some of the more basic and standard ones, we use a set of tools that we've written in MATLAB and Perl, and I'll show you the output of those in a moment. And we're also developing a way that we can deliver and visualize data via the web. And this is a challenge because unlike other platforms, we're frequently interested in the sequence level data, and we often get hundreds of thousands of sequences for these experiments. And so this is a direction that we're moving towards to try to figure out how to enable that. And on this side, we have the metadata tracking. So we do everything from clinical data, data about the draw, cell counts, information about the wet lab process, and information about what parameters were used for the analysis and which samples and cell subsets are included in the analysis. And also, I have R listed here. We haven't done anything in R, but the way we've designed this, it would be trivial to develop a tool that can interface with this database and encapsulate some sort of analysis or some standard kind of question you'd want to answer in R if you wanted to do that. We just work in MATLAB. This slide shows a couple of examples of the different visualizations that we can generate using this platform. You can answer questions. I'm not going to go through this in a lot of detail. I just want to give you a sense for the kinds of questions you can answer. The top one here is a heat map showing the distribution of V and J gene usage across many samples. So the rows are different individuals, the columns are samples, and it's a way to assess the similarity of the distributions of the gene usage between samples and sample types. So for example, we have lupus and vaccinated samples. And then on top of that, you can overlay information about lineage size. So you can ask whether what are the largest lineages in these samples, and overlay a circle to show where they are and which V and J gene usage they have. And all of these visualizations are tools that are, except for the Circos plot, are generated immediately from the databases. We can answer questions about clonality. In this case, we're showing 4 cell subsets: naive, memory, plasmablast, and plasma cell. And we're ordering the lineages based on size. So as expected, the naive cell population is highly polyclonal, consisting of many small clones, whereas the plasma cell are very expanded. And this is a vaccine sample for 7 days. And on top of that, we're coding each one of these lineages using barycentric coordinates, namely color. So we have 3 different colors for IgA, IgM, and IgG, and you can see that the distribution of sequences within each lineage is coded by a mixture of the 3. And as expected, the naive are mostly IgM. We can show plots related to the mutation rate for different cell subsets, different isotypes, and different samples. And so this is a way to visualize the mutation histograms of 40 different plots in a very small area. And essentially, it's using a heat map instead of a bar chart histogram to be able to do that. We can have many ways of quantifying diversity. So diversity means whether you have a polyclonal distribution or an oligoclonal distribution with few dominant clones and many and fewer smaller ones. We can also track lineages over time. This is an example. These are clonality plots for plasma cells at 3 time points during a lupus flare and 5 weeks and 8 weeks after a lupus flare. And so here again, we're ordering the lineages from smallest to largest, and you can see that there's a couple of expanded clones here. And what we've shown here is we've asked, what are the top 10 lineages that are annotated with the V gene for VH434, and are they present in the subsequent samples? VH434 is interesting because it's implicated in autoantigens for lupus. And then below these plots, we can show the sequences of the CDR3 nucleotides as heat maps, and we can show that we find the same sequences and mutated sequences at all 3 time points. And finally, in the center is the Circos plot. And what we're trying to show with this is to ask whether lineages span different cell populations. And in this case, we've set it up so that we have lineages from plasma cells. So each arc here is like a single plot here. And we're asking whether the plasma cell sequences, do they connect to lineages in the switched memory population or the naive population for both plasmablast and plasma cell. And so the fact that you have a ribbon going from this very large lineage here to a large lineage here indicates that there are sequences that are called the same lineage as a result of the rules that we applied that I talked about a moment ago. So to finish up with the sequence, yes?
So in this data, if I look at that central circle, I see very few lines going from plasma cell to plasmablast or vice versa.
That's correct. So in reality, it's quite connected. We just eliminated those connections for the visualization because otherwise it would obscure what we were trying to look for. I mean, in reality, what we often do now is we'll combine the plasmablasts and plasma cells into one population analytically after the fact and then just show this kind of plot. So next, I'm going to switch gears a little bit and focus on an older platform, namely flow cytometry. And this is the platform I worked on when I first got to Rochester. And there were 2 projects that were generating an enormous quantity of flow data. And these were 12, 16 color flow machines and multiple flow panels. So we were measuring different B cell panels and T cell panels. And this was for hundreds of individuals and multiple time points per individual. So it was a very large data set. And then in another project we worked with, we were looking at high resolution flow cytometry sampling for vaccine response. And so one goal was to come up with a workflow that would facilitate dealing with this large data set, because the traditional way to do it is this program called FlowJo or something like it, where you have manual gating and you'll take a data set, and the data set is multidimensional in that you have multiple markers on the cells, and you can only look at 2 at a time in this method, and you you recursively subset your samples like this, and we're only looking at, in this case, of B cells, we're looking at IgD versus CD27 markers. And this is what's used to separate the B cells into 4 canonical populations. But this is a manual process that involves putting gates on sets of cells, and it can be tedious and very impractical to use for large data sets, and is also prone to bias because it involves human intervention. So I work with Martin Zand at University of Rochester. We were trying to come up with an environment to try to solve this problem, and we were using a program called Flock, developed by Richard Sherman. And so we incorporated that, and what Flock does is it takes the event level data, so that's shown here. Event level data is the raw data produced by the cytometer. So each row is a cell passing the detector, and each column is a marker whose signal is being detected. And this is, for those of you who work in flow, this is what comprises the FCS file. What the clustering does, instead of looking at 2 dimensions at a time, we can cluster the events, and Flock uses a density-based method of clustering into using all the dimensions. The dimensions are the markers in the cell panel, and we might come up with 25 clusters in this multidimensional space. And then, so now we have a bunch of subsets, and then the next challenge is to figure out what those subsets are. And so what we've developed is we built a prototype of a database of markers and cell populations taken from the OMIM panels for B cells, T cells, dendritic cells, and all sorts of other panels. And we built a database, separated them by markers in a way to have a web-based platform to map the clusters to cell populations based on which, so these clusters are characterized by expression level, these centroids are characterized by expression levels of markers. And then through this database, we had a semi-automated approach to guess which populations these unannotated clusters belong to. So the goal was at the end to get to a point that you would get to with FlowJo, where you would enter your FCS data and get your populations and their relative abundances in the sample. And this plot here shows a way to visualize the clusters. This is an experiment where we had 11 time points for a vaccine study, so 11 successive days. And this is a method called multidimensional scaling, where we take 8-dimensional data. Each dimension is a cell marker here, and we reduce it to 2 dimensions to try to preserve the relationship in distance from the clusters. And then these glyphs on this plot show the relative abundance of each of the markers. And from that, we can see that these represent antibody secreting cells, these are memory cells, and these are naive cells down here. So that this is multidimensional scaling. It's very similar to the principal coordinates analysis people use in microbiome research. And then finally, this visualization shows a heat map of those relative abundances. In this case, it's 6 markers. So each row here is a cell cluster. Each column is a marker or dimension. And then these plots represent the tightness of the cluster. So silhouette is a way to characterize the nature of the cluster. So for example, these are very well-defined and tight clusters. These are more diffuse clusters that may overlap. And then finally, we take one sample or one individual and plot a composition plot over time to show the B cell composition for all of these subsets. And so you can see at day 7, there's an expansion of the plasmablasts and plasma cells.
So I assume that this data works well if you have a distinct population. But what if the populations are overlapping? For example, if you have a common population for CD20 positive, which is also CD19 hazy, it's not complete positive. How will that, this plot will work on that sort of data?
If you guys, when you have a question, wait till we get, because the people online can't hear you, and then the recording as well. So
I can repeat the question.
I think you can repeat it, but just for the rest of them, if you just wait till we get a mic. Thank you.
So the question was how well this method can distinguish between data that's very diffuse. So often when you're looking at the flow plots, you get, rather than see distinct clusters, you get a smear points. And the answer is that it will find clusters when there may not be distinct clusters. And so although this is a reduction in the dimension space here, there's really 8 dimensions in this data. If you could see in 8 dimensions, you would see that these 2 clusters are fairly close together. And you can actually see that up here because these are the relative. So for example, these 2 right here have fairly similar expression patterns. They're distinguished by a slight increase in the second marker and a slight increase in the third marker, but otherwise they're very similar. And so it's not perfect and it's not going to capture, it's going to assign each event to a cluster, and that might not be correct because, as you say, the data is not always distinct. Okay, one more question?
It's an extension. So it's an extension of the previous question. So let's say, I'm assuming that this data works well if you know what the population is. For example, you have defined T cell as CD3 positive. What if you're trying to decipher new populations, new markers? How would you, basically, when you don't have any particular defined set of definition, then how would this program work for that?
So the program is, up till this point, the program is agnostic to markers and how they cluster together. But this program is dependent on which markers you enter in your flow panel. So you only have this to work with. And so if you divide your expression level into 4 quantiles, for example, and this looks like about 8 or 9 markers, that gives you a lot of possibilities of combinations. And so you can get that data out, you can look at the centroids, and it may be a pattern of markers that don't correspond to any known population that have been annotated. And so that's how you would have to go about doing that. Does that answer your question?
Yeah.
Okay, thanks. Okay. One other thing I want to point out, it's a slight shift in topic here, is I want to mention the data management component for this. And I'm going to use flow cytometry as an example because this is a prototype I built about 5 years ago. And this was to deal with the problem of hundreds or thousands of FCS data, or rather the output of the FlowJo data, cell subset frequency data for hundreds of people and multiple time points. And it became a really challenging data management problem. And it was very difficult to analyze this because everything was in different places in different formats. And so what I did was built a database to unify all of this. And I also provided a front end. And this is an example of the kind of data management platform that can tie in closely with analytical tools. And this would be very useful for a variety of different platforms. And so if you have a front page here, you can navigate between subjects, which can give rise to many draws, which can give rise to many flow runs, different B or T cell panels. Each flow run can go to different analyses, which we don't typically do. Then we get counts and frequencies, and we could have other immunological data. So now we have everything in a structured environment, and everything is in the same place together. So you can ask very specific questions about things like, show me all the patients, this is a lupus study, so show me all the patients with high disease activity index that have a plasmablast frequency over 20%, something like that. And those kind of questions are very easy to ask. You can do it either by the web interface, or you can do it by plugging in the MATLAB or R tool directly to the database. And that's a very useful approach when you're working on a paper and you want to have figure provenance. That's a really useful strategy to use, because you can have a script, you have a data source, and you can have a record of everything that you do to it. So this is an example of just a subject page where you have the information about the subject, and this is everything that has happened to that subject longitudinally. So you have the subjects that has 3 draws. This draw had 2 flow panels. This flow panel had 2 analyses, and so forth. And everything's hyperlinked, so you can click on things and it'll get you to the appropriate page. You can actually get to the data itself for both the count and the frequency. So finally, I'd like to acknowledge my coworkers. So for the Repertoire sequencing, this started about 5 years ago. I worked with Chris Tipton and Iñaki when they were both at Rochester. And Chris came to me with some initial 454 data. He had 3,000 sequences, and he needed to cluster them. And there were no tools at the time to be able to do that. So we started this collaboration, and it has evolved into the current pipeline that I showed earlier. And then for the flow cytometry workflow, that was in Martin Zand's lab at Rochester, and also Melissa Trahan did a lot of this software development for this. And finally, I want to give my contact information. I'd love to talk to any of you who are interested in discussing your own data needs or analysis needs. I also want to point out my analyst, Chris Fucilli. He's RIT Bioinformatics master's student, and he did a spectacular job building all the software for this pipeline based on my less than elegant beginnings several years ago. He's really taken analysis for this, and he also did a lot of the software for the flow analysis as well. So these are our email contacts and our phone numbers. This is my Rochester number, and this is my office in Bevel. So thank you very much.
We still have a question. Oh, here too. I want to let you know that we have a 30-minute panel discussion after the first 4 speakers. So don't worry if your question doesn't get answered. So we're gonna start. Yes.
So I have a question about the first part of your talk when you talked about your lineage assigner, and so it seems like, I mean, you mentioned it's really dependent on the rules that you define yourself. Mm-hmm. So obviously your rules then set the stage for all of your data analysis following that, right? So in this particular case, so it's kind of related to this previous question. How did you define your rules in this particular context? And I can imagine it'd be it's different depending on whatever your initial, your source data is coming from, like what cell type and what your question is experimentally. But then how you envision your approach being similar maybe to other people's approaches. I'm just trying to think about this in how the lineage assigner sets the stage for all of your data.
Right. That's a great question. So this is a common approach used by other groups, and oops. So we're talking about this area right here. So we know that they should share the same V and J gene. So that one's not really ambiguous because a antibody secreting cell derived from a naive cell is not going to change a recombination after it's a mature B cell. So really the parameters we have to work with are the similarity between the CDR3 regions in this context. And some people, so we're using straight homology. So we've tried the sensitivity analysis where we vary the degree of homology to try to see how drastically it changes the lineage assignment. And the way it's really kind of a chicken and egg thing, and it's hard to validate. So what we'll do is if we start out with say 70% homology, we can look at the sequences in large lineages and try to see if it actually makes sense. And right now what we're typically using is 85%. We could also look at the junction regions because the sequences at the junction regions would be a proxy for being part of the same lineage, although they can be susceptible for mutation as well.
For the time's sake, so let's hold your question to the 30 minutes panel discussion. So we want to give each speaker a fair amount of time to talk about their thing. And we have 2 panel discussions this afternoon. So make sure your questions get answered. So our next speaker.
So this is forward back, and that's the.
Is Chong, and he is also a new member in the Informatics Institute, and he's home based in genetics. And he has a lot of experience in sequence analysis, high throughput sequence analysis. There are so-called short reads, long reads, and also genotyping, genetic variation analysis.
The first one.
So
the first one. Yeah.
If you have this type of a question, and then you can ask him here or afterwards.
Okay.
You're all set?
Sure. Thanks for the introduction. I appreciate to have the opportunities to present at the mini symposium for forging new bioinformatics collaborations.
Activate the microphone, please.
Oh, sorry. I think I have.
Oh, just speak louder. Yeah, stand behind the podium and then louder.
Okay, sure. Okay, I will be louder. Yeah. I have been here for just 2 months, so it's my 2-month anniversary, and thank you. So today I would like to talk about my work on structural relation analysis using Illumina short reads and PacBio long reads. So what is structural relation? Structural relation is a type of variations that have structural changes both between and within chromosomes. It can simply be classified as deletions, insertions, inversions, duplications, and translocations, etc. Note here, I use the word simply. Indeed, structural variations can be very, very complex. A cancer genomics analysis hypothesis by Sanger Institute proposed the hypothesis for named chromosome synthesis. It challenges the traditional view of stepwise accumulation of mutations during cancer evolution. It hypothesizes that a catastrophic event happening on one chromosome causes hundreds of and thousands of rearrangements. The aberrant repair of the segments can lead to cancer development and progression. This is a sample from TCGA, and this is a colorectal cancer sample. On chromosome 15, there are 239 rearrangements on this locus. So this phenomenon is very interesting, and there's quite a few follow-up studies in terms of both the similar observations or some underlying mechanism studies. So structural variations are so important that we should comprehensively characterize all structural variations. However, due to the technology limitation, we cannot achieve the goal easily. So detecting the structural variation is still very challenging. The first problem, because the sizes of structural variation have a wide range, it can vary from 100 base pairs to several mega base pairs. And different types of structural variations have different mapping signatures. And the structural variations can be complex, as mentioned, the chromosome synthesis. And the NGS rates are usually very short, 100 or 120 or 150 base pairs. And structural variation breakpoints embedded in the short reads are usually very difficult to align to the reference genome. And if structural variations can be heterozygous, so the heterozygous structural variation can make the useful reads rare. In cancer genomics, the purity or subclones of the tumor sample can make the useful reads even rarer. Currently, there are 2 major types of structural variation detection algorithm. One is resequencing based. The other is de novo, whole genome de novo assembly. The resequencing based methods include read pair analysis, read depth analysis, and split reads analysis. All these resequencing based methods depend highly on the mapping results. But we know the structural variation, if embedded in the short reads, is difficult to map to the reference. So the split read methods, you want to split into 2 smaller parts. So it's even harder to align to the reference genome. And the read depth method can only detect copy number gain or copy number loss. And read pair methods cannot achieve an exact breakpoint resolution. And usually the false positive rates are very, very high. And we all know that de novo assembly problem is a challenging problem per se. Because of the sequencing errors, the heterozygous and the repetitive sequence in our genomes. So it's really hard to to do a de novo assembly to generate a contig to cover the breakpoints. So current methods have a limitation in terms of both sensitivity and specificity. To achieve a better sensitivity and precision, we developed a novel break. We developed a method called no break. No break takes full advantage of a k-merge strategy. What are k-merge? K-merge are all short or all length k substrings of a sequence. We observe that if a breakpoint exists in a read, the breakpoint should exist in the subset of the k-merge of that read. We call those k-merge novel k-merge. So no break try to discover those novel k-merge and the reads associated with those novel k-merge. Here's the workflow of novel break. No break starts from a sample reads. It can be a tumor reads. First, it will be chopped. The short reads will be chopped into k-merge, and we will index the k-merge into a hash table or a dictionary data structure. Then we will filter against the reference. After filtering the reads with somatic mutations, the k-merge with somatic mutations, with germline mutations, and the sequencing errors will be left. Here we adopted a counting bloom filter data structure to filter out the low count error-prone k-merge. So after filtering, after this counter bloom filter, we will only get germline or somatic novel k-merge. If there is a normal sample, we can further filter against the normal sample to retain only the k-merge that present in the tumor samples. This is the somatic novel k-merge. Next, here's the hidden structure. If there is a breakpoint, for each breakpoint, there's many novel k-merge covering the same breakpoint. And for each novel k-merge, there are many read pairs sharing the same novel k-merge. So based on this relationship, we can cluster the read around the breakpoints into a cluster. And after clustering, we will do a local assembly to generate a contig, represent the original sequence that cover the breakpoints. Next, we will align the contigs to the reference genome to infer different types of structurations. Finally, we will score and output the VCL in a VCL file of the structuration predictions. We demonstrated the performance of a no break by participating in the ICGC-TCGA DREAM Challenge in silico data. The DREAM Challenge is a worldwide competition. The purpose of the challenge is try to identify the most accurate algorithm for identifying mutation calling algorithms. And there's a total of 4, officially there are 4 sub-challenges. No break achieved the highest sensitivity for the first in silico data, but the overall, the precision is a bit low. So the overall, we ranked in the 4th place. After that, I developed the scoring algorithm to filter out the false positives. So for subsequent saturation sub-challenges, no break consistently ranked in the 1st place. Since the third synthetic data set, indels were introduced. No break can be applied to discover somatic indels as well. We participated in this indel challenge, sub-challenge. No break ranked in the 2nd place. And we tuned the parameters and try to improve. In the 4th sub-challenge, no break achieved the highest, ranked in the 1st place for indel discovery as well. This is some pictures that I attended the annual DREAM Challenge meetings. And the in silico data 3 is the most challenging data set because there's a 3,000 of structurations introduced, including deletions, insertions, inversions, and translocations. And there are some clones in the data set, and there's indels simulated. So this data set is the most challenging one for structural variations. And this figure shows the top 3 performers of this challenge. All the dots represent all the submissions of 3 top performers, daily, meta, and no break. The stars indicate the best result of each method. As from the picture, from the figure, we can see that the precision of all these tools are very high, but no break outperforms the other tools, mainly due to its high sensitivity, especially for insertions. No break actually can identify 100, which is 4.3% of true ground truth insertions than meta, and 120 more true events insertions than daily. So that's why no break is better than the others. Why no break is better? Because no break can use more reads. This is an illustration. For alignment-based methods, if there is a structural variation, short reads are usually difficult to align to the reference genome, and some of the reads will not be mapped to the breakpoints, and some will be partially mapped, and some will even be wrongly mapped to the breakpoints. But the k-mer-based clustering method, as long as there's novel k-mers, we can cluster as many reads as possible and to reconstruct the original breakpoints. Another very important factor for structural variation analysis is the breakpoint. Breakpoint accuracy is important for subsequent experimental validation or subsequent mechanism analysis. I compare the 3 top performers of the for the breakpoints, and again, no break. Almost 99% of the breakpoints fall within the -2 to 2 base pairs compared to the ground truth, which is the most accurate, precise tool for breakpoint accuracy. Next, we benchmarked the performance of no break based on the real data, real cancer data. One data type is the cancer cell line, is COLO-829, is melanoma cancer cell line. And previously, there are 2 papers identified and validated 48 experiment validated 48 of structural variations. We use these 48 as ground truth to identify the sensitivity and the specificity. And usually for bioinformatics, for users, the default parameters are very important because most of the biologists do not know the algorithms or have little experience in bioinformatics. So we compare the default parameters of these popular tools. And no break achieve the highest sensitivity with the minimum total cores. And for those novel cores, we further validated 9 of the events. And you can see the other tools cause more than 1,000 or even tens of thousands of total cores. So implies there's a lot of false positives. I also analyzed the TCGA samples, breast cancer samples for 22. Previously, there's a paper published by Genome Research. There is a tool called Integrate, identify 120 gene fusion using both whole genome sequencing data and the whole transcriptome sequencing data. And no break can identify 104 of them, with 10 in the low quality regions. The overall sensitivity is 87, or including the low quality would be 95. So the sensitivity is very high. The sensitivity of no break may be even higher because the integrate has a false discovery rate of 19%, and no break only use the whole genome sequencing data. The paper was published in Nature Methods. And although, despite no break achieves very good sensitivity and specificity for saturations on the Illumina short reads, we can see there is still a gap. There is still challenging to perfectly discover saturations. We can see in dreams in silico 3, the overall sensitivity of a no break is only 80%. So is there 20% of sensitivity gap? On one hand, the challenge is really, the simulation data is really challenging. On the other hand, yes?
Can I ask a question? You showed in earlier slide about 120 gene fusions. Which cancer that was?
Yes? Oh, sorry, I cannot.
Cancer that gene fusion you found?
Oh, it's breast cancer, breast carcinoma, breast cancer.
How many of them are validated by any other biological methods?
Actually, no. We just use the integrate methods, integrate results as the ground truth, because integrate both whole genome sequencing data and whole transcriptome sequencing data. This paper was published in Genome Research. They have a cell line data for validation, and they analyzed additional TCGA samples for further comparison. And they haven't validated the TCGA sample, but there is an estimate for the sensitivity and the specificity based on the cell line data.
Yeah, I was asking how many of them are like recurrent gene fusions, for example, and how many of them are actually has meaningful consequences in cancer. Like many of them may be passengers, right?
Sure.
Way you can, because in breast cancer you don't have any gene fusions.
Yeah, that's a biological question. So currently I just focusing on the methodology. So yeah, that's my interest, but I haven't compared the biological meaningful of these gene fusions. So there's still a gap for saturation detected using short Illumina reads. The third generation sequencing, because of its longer reads, typically the read length will be tens of or even hundreds of base pairs. So it's quite promising for detect saturations. The PacBio use a enzyme reaction to read through the insert template, and it can read through the template back and forth, and to generate a polymerase read. The polymerase reads can further be processed into sub-reads. And by calculation of the sub-reads, it can generate a consensus sequence of the sub-reads called circular consensus sequence, CCS sequence. The problem with PacBio data is its high sequencing errors. The error rate, overall error rate would be 15%, and there will be like 14% of the error will be indels, and 1% will be SNVs. So it's very challenging to analyze PacBio data. And with the collaboration with Dr. Lapsky, James Lapsky at Baylor College of Medicine, we are working on the project that try to identify, to study the mechanism of copy number variations on a low copy repeat rich genomic region on the chromosome 17. And there are five large low copy repeats with similarities over 98%. And besides, within those large low copy repeats, there are some inverted or directed short low copy repeats with similarities higher than 99%. So it's extremely difficult to identify. All the breakpoints are lying in the repetitive regions. So it's difficult to identify using short reads. And for this region, the copy number gain will lead to a syndrome called PTLS syndrome. And the deletion of the region will lead to a syndrome called SMS. And there can be a non-recurrent and recurrent copy number in this region. Dr. Lapsky group capture of the region using an experiment, capture about 7 megabytes of this region, and both Illumina sequencing, both Illumina and PicoBio platforms were applied to this DNA and generated both Illumina data and PicoBio data. For the Illumina data, we tested several popular sort variation detection tools. Sadly, we hardly find any exact breakpoints for these samples, these 22 samples. No break is the best one, but it can only identify 4 of these samples. So I developed a pipeline to analyze PicoBio data. The PicoBio raw data can be processed using the PicoBio pipeline as to a sub-reads or a CCS reads. And the CCS reads or sub-reads can be assembled into long contigs. During the assembly process, there is a step to correct the error reads of the PicoBio reads. There is also a corrected reads. So the pipeline of PicoBio analysis, the long sequences include sub-reads, CCS reads, corrected reads, and contigs. And I modified the parameters of BWMAM to adapt the alignment to long sequences and to generate the SAM file. By passing the SAM files, we generate a raw call size. And by clustering based on the adjacency of the breakpoints, we can generate a call group for these breakpoints. Finally, we will merge the results and report the final calls based on the number of reads or the ZMWs. And interestingly, using these methods, we identified exact breakpoint for 19 of these 22 samples, and we can also validate them. Most of the non-recurrent common number variations can be used, can be identified exact breakpoints using PicoBio data. But there is also a challenging for recurrent rearrangement. For example, this sample, there's from array CGA, there is a clear deletion involved there in that region. But for PicoBio, we tried and we cannot find the exact breakpoint. The estimated breakpoint is at these 2 large low copy repeats, which is 180 kilobase pairs. So it's really difficult to identify the recurrent sequencing. We are still working on that. And in summary, we present a novel method for discovery saturation. It's called novel break for short reads analysis. It uses a k-mer filtering strategy. It can cluster and local assembly of the reads. By benchmarking using the DREAM challenge simulation data and the real cancer cell line and the real TCGA patient data, it has a higher sensitivity and specificity compared to other tools. And we also provide a solution for resolving the saturations involved in low copy repeats by using the PicoBio data analysis. The novel break work was supported by Dr. Ken Chen and Dr. Gordon Mills at MD Anderson. And I would like to thank Dr. Ming Gao in Baylor College of Medicine for the experimental validation and testing of the algorithm. And Dr. Lapsky, Claudia, and Christine for the PicoBio project. And I involved in the 1,000 genome projects and tested my pipeline using lots of their data. And thank the DREAM team, Paul, Anna, and Kostova. And I would like to thank Informatics Institute, Jake and Jim for their supporting my research. And I have currently working with Dr. Bob Kinkley with a very exciting problem involving in the 10x genomic data. Finally, I would like to do an advertisement. My live launched on March 1st. We're pretty new. And the mission of our live is try to make sense of biology using bioinformatics. our research is not limited on structurations. We have broad interest in the genomics field, and we are interested in developing new algorithms for sequencing data analysis, and we are interested in function and mechanism of rearrangements. We are interested in disease genomics, like cancer genomics and Mendelian disease genomics, and the collaborations are welcomed. And we have currently we have a few openings for postdoc and graduate students and undergraduate students. And you feel free to contact me at ZC2NG. And thank you very much for your time. And I would like to take any questions.
Yeah, that is a good point, making sense of biology. Because when you send a list of 300 gene fusions or 500 mutations, doesn't make any sense because you put through some filters, you'll get anyway. But a lot of the low-hanging fruits have been identified already. So what else you can do in terms of biologists? I think that is a major question for cancer biologists. Let us say, how do you look at that instead of let us say taking TCGA data and finding another 500 gene fusions or mutations? What kind of methodology you would use to help biologists?
You mean for biologists?
Yeah, because I mean any tools or web portals you use for identifying gene fusions, you end up getting a lot of them. But how one can filter it to make sense out of those data? That is, I think, a important issue moving forward.
You mean the application of the structuration or this type of results, right? In the biological studies, right?
Like, let's say ERB2 amplification in breast cancer. It's very well known, let's say. And then what else can be done? If ERB2 is amplified, what is downstream? So for that,
you mean
from genomic sequencing?
Yeah. You can
network analysis and.
Yeah, the first thing is you can characterize those structurations in different type of diseases or whatever genomes, right? The second thing that you can use these structurations is like you can study the mechanisms, and you can put a cohort of the studies. The mechanisms of structuration are very fantastic because there's still open questions. There's different mechanism for generating the different type of structurations for recurrent or non-recurrent ones. So it's straight. Currently, technically there's a difficulty, and biologically there's lots of questions need to explore. And yeah, that's it.
Please hold your question. So to our panel discussion. So our next speaker. So our next speaker is Ming Gao, and she works on this hot thing called the single cell, and you guys probably have heard about it nowadays. And so single cell sequencing, single cell sorting, and doing various things. And she, before coming here, and she used to run this single cell core at Baylor. Right. Is it Baylor?
Yes.
All right. So she knows all about this single cell. So if you think about this technology, come to talk to her and ask her to help you.
Okay.
You just have to find it.
Okay. Hello, everyone. I'm also the new one. I'm here also only for 2 months. Thanks for giving me this chance to introduce my works on single cell.
Louder. You need to speak louder. They can't hear you.
Okay.
Yeah. The microphone won't amplify. It's just for the online people.
Okay.
Right. Speak louder.
Okay.
Just talk to the guy at the back. Just talk to the people at the back.
Okay. I will introduce my works on single-cell genomics and data analysis.
Or you want to stand there to talk because they still can't hear you.
Maybe you can come closer and then talk. Maybe they can hear you back there.
Right, right.
Okay.
Do we have, I don't know. We don't have, okay, just talk louder.
Just
Okay.
scream at the top of
Yeah,
the voice.
scream, talk to Steve at the back, like, so that he can hear you.
Okay. I can do the.
There we go.
This is not
No, you gotta
working.
work two. One's for recording and then this will be for in here. Just leave that. Just leave that. Just leave that in your pocket. Just leave that in your pocket.
Okay.
Yeah.
There you go. We're gonna put one on you. We're gonna put the second test one too. Put that one on you as well. And just put that one in that pocket.
Yes, it works.
Yeah, you're gonna have to get it, unclip it. You're gonna have to get it way up by your mouth.
It works.
No, I'm gonna figure it out. Guys, I'm sorry, this isn't my room. I'm just helping out. So I'm learning as we go as well. You can hear it, but you can't hear it. So let me see what I can figure out. Hang on.
Try again. Say something.
Yes, it works. Yes. I can do the next generation sequence data analysis, include the whole genome sequence, whole exome sequence, and I-seq sequence data, and bisulfite-seq and chip-seq. Especially, I'm good at single-cell sequence data analysis, such as the single-cell whole genome sequence and whole exome sequence. Also, the single-cell I-seq data and a small amount of cells I-seq, and the single-cell bisulfite-seq and a small amount of cells bisulfite-seq. So I'm now open to the collaborative project for data analysis for NGS data analysis and single-cell sequence data analysis. As we know, the fundamental unit of our organism is the single cell. Each single cell or cell type has distinct functions. Research on the single cell can provide great insight in our understanding for the mechanism of the human disease. With the development of next generation sequence, many more and more studies focus on single-cell sequence-related research. And this figure shows there are growing numbers of publications on single-cell sequence-related research. Single-cell sequence impacts diverse areas, includes the neurobiology, cancer biology, immunology, microbiology, and so on. In our neural system, we know there are many different cell types, and use the single-cell sequence technology, we can study the mosaic genomics of individual neurons and compositions in the brain, or follow the genetic variants during the fetal development, or developed the target therapy for neurology disease for specific cell types. For cancer biology, the accumulation of somatic mutations can lead to polyclonal tumor, and in which different clones may be respond differently to treatment. So use the single cell sequence technology, we can find the evidence for models of cancer and to infer timing of the mutation and the drivers. and also evaluate the effectiveness of the targeted therapy. In our immune system, there are also a lot of different kind of cell types involved in this system. So use the single cell sequence technology, we can investigate the heterogeneous transcriptional response in immune cells after antigen activation, or identify the unique gene expression pattern and splice virus in T cells or B cells, or find out the interaction between the immune cells and the tumor cells. Now I show this. This is a workflow for single cell RNA-seq or genomics DNA-seq. The single cell came from the solid T cell or from the cell suspension. We can use the different method to isolate the single cell. It can be used the micro manipulate or cell sorting or enrich, or tube or microfluidic based method to isolate the single cell. After we get the single cell, we can extract the genomic DNA and use the different method to amplify the genomic DNA. Or we can get the iron, use the RT-PCR to get the amplified the CDA, and then prepare the sequence library and do the sequence. Finally, do the data analysis for the single cell data to find out the allele drop-off, copy number variation, or identify the different cell types. Now, I will show some case studies for single cell sequence related research. First, I want to show the case study for identification of different cell types. The sample I use is the mouse uterine cancer cells. I use the Flowdam C1 single cell system to capture the 16-7 single cells, and then amplify the CDA and prepare the library for do the low coverage RNA sequence. After get the sequence data, we can identify the different cell types of the mouse uterine cancer cells. When I get the data, the sequence data for the single cells, I use the in-house RNA-seq pipeline to analyze the data. First, to do the quality check and trim the reads, and then mapping the reads to reference genome. We use the top hat tool and use the cufflinks to get the gene expression list for each single cell. Summary the result for the data analysis. I get the QC report for the single cell RNA-seq samples. From this report, we can see the percentage of MI level is about 17%, and the 5' bias or 3' bias is low from here, and the mapping ratio is about more than 17%, and the total gene detected for each cell is about 6,000. So the quality of the sequence samples for single cell is good, and it's useful for further analysis. This is also the QC report. And for the further analysis, first I identify the outlier for single cell RNA-seq samples. The blue ones indicate the good quality ones, but this red one is the low quality ones. So before I do the further analysis, I need to remove these samples. I plot the PCA figures for single cell ISEQ sample, we can see this single cells this single cells different from this population. And I also plot the heat map for the single cell I-sequence samples. We can see this population is significant different from other single cells. Our collaborator labs have used experiment to verify these different cell types. The papers include this past work will submit soon. Another case study is for to test a new method of single cell isolation. Our collaborator labs developed a new method to pick out the single cell. Use this new method to pick single cells from breast cancer cell line, and use the smart stick to manually do to amplify the CDA and prepare library and do the low coverage ion sequence, and to test whether this method is worked for pick a single cell for ion-seq. After getting the sequence data for the samples, I also use the in-house pipeline to analyze the data. Finally, get the gene expression list for every sample, and I summary the results. Get the QC report. We can see the mapping ratio and the percentage of MI level and the total gene detected is good. And for the 5' bias and 3' bias is low. And also from this figure, we can see that there is a high correlation between single cells and bulk cells. So this indicated this method is good for pick single cells for I-seq. This work has published and the paper has online now. Another case study is for clustering. This shows the different cell classes in mouse retinas. It contains photoreceptor, horizontal cells, bipolar cells, M-corin cells, ganglion cells. For ganglion cells, it only contains 1% in retinal cells, so it's very difficult to isolate this kind of cells. Our collaborator labs have isolated ganglion cells, and we use these ganglion cells as a sample and use the 1-gene ICLH single cell system to capture 509 single cell and amplify the CDA, prepare library, and do sequence. And wanted to clustering the mouse retinal ganglion cells and identify the different cell types. After I got the single cell sequence data, first I used the 1-gene developed pipeline to get the gene expression list for each single cell, and then do clustering for the retinal ganglion cell. It can identify these 6 groups. For the further steps, I will detect the marker genes for each group. For the next generation sequence data analysis and single cell sequence data analysis, we have in-house pipeline, include SNP and EDL calling pipeline, copy number variation calling pipeline, and I-seq, bisulfite-seq, and chip-seq pipeline. Also, we have the computational infrastructure for data analysis. It contains multiple nodes and multiple cores, and has high performance storage. Also, we combined the open source Galaxy to different workflow motion. It make it easier to share the new tools and workflows. Finally, thanks Dr. Chen giving me many suggestions on data analysis. Thanks Dr. Semino recruit me here to work here, and thanks Dr. Chong giving me many suggestions on bioinformatics, and thanks all the members in Dr. Chen's lab, and also thanks my former mentors and collaborators in Baylor College of Medicine and Houston Methodist Research Institute and in MD Anderson Cancer Center. Thank you. Okay.
Hi, Min. So in your experience, how many single cell analysis do you need actually to validate a result? Is just one single data from one cell is sufficient to validate it? Or do you need multiple cells to validate the same results? Because in one of your slides, you showed data from 509 single cell analysis. So in general, what is the least number of cells you need?
It depends what do you want. If you want to identify the different cell types, it's normally you need a sequence more than 10,000 cells to clustering. For 100 cells is not enough for clustering. If just I saw in the case study, it's only use 500 cells. It's only identify the six class, so it's not enough. So normally it's use 10,000 for clustering. But when I also only use 2 or 3 single cells sequence data to test the different new method for pick out the single cell technology. So it's only 3 samples, it's enough. But also we show for the FlowDAM C1, if I use less than 100 cells, it's very likely we found the new population use less than 100 cells. So it depends on what you want.
I have to ask you to hold your question again. I'm so sorry. But you will hold on to your microphone, and then you become the first one to ask. So our next, I forgot about this. Sorry. So our next, I'll pass it to you afterwards. So our next speaker is Jake Chen, and are you ready? Yeah. And not yet.
The slides.
And he's a professor in the genetics department and also the chief bioinformatics officer in the Informatics Institute. I think that he has been here a little bit longer than the other 3 people, not too much, as Jim said. He is going to talk about, let's see what he is going to talk about.
In the presentation folder.
Transforming omics data and visual storytelling. And I know he has been doing really a lot of work on establishing this system biology tools. And if you have problems and you want to connect a lot of different type of biological data, annotation, gene network, this type of thing, and this is the guy to talk to. He may not talk about all of those, but he has really nice tools for doing system biology. Jake?
All right. Thank you very much. Am I loud enough for people in the back? All right. Great. So I'm going to talk about storytelling today. Or actually, I could have a subtitle that has lots of the familiar object, sand, castles, and I'll keep some mystery. So I like to actually put up my messages up front. I know a lot of you are not bioinformaticians, so you can't tolerate formula. I remember 20 years ago, I couldn't do that. So I'm going to send you some abbreviated message in case you're completely lost and this is the things that I'm for you. So one is, I think the omics data, we talk a lot about them, we collect them, we like to actually cherish them, publish them, but they're really less useful unless you satisfy these two criteria. One is that the data has to be curated, linked, and aggregated with contextual knowledge. And those knowledge may be in your brain, and you learn for so many years for your PhD, but for data scientists, we get them from public databases. And if you really want to do some super cool stuff, you need to have new transformations, okay? And then our goal to construct models from the data is really to make them understandable. It's not to actually make you scared away and then say, yes, I bow to your power. No. And so we really want to understand the complex biology. And I think the future bioinformatics science requires not only all of these engineering skills, but I think a little bit of a data artistry, and I'll talk about today. And so these slides are probably everywhere, and you are very familiar. And essentially, the idea is that the omics and computing has been very pervasive in the practice of medicine today. And in our Informatics Institute, we really want to embrace computation and the omics so that we can keep track of a persons from birth to death using all of the omics data to really help them improve their health. And also on the research side, do multidimensional omics, genomics, environmental studies, social biosensors. On the other side is different microbiome exposome to capture all the data so that there's no black holes. And then we want to capture them periodically over multiple time frames. And to the point that we can now talk about precision medicine. According to Dana-Farber's definition, it's really simple. We bring all of these omics studies, we make some discovery on which patient have which mutation, and then we pair them with drug. If a patient have different mutation, we use different drug. If the patient have different disease but seem to arise from the same mutation, we recycle the drug, right? But I think the key is there's an analogy. So we get lots of data. We don't really get the data that really pinpoint the right solution. So we're at the digging side. And I think there's a perfect analogy of the digging side, which is messy, not obvious to the complex disease biology that we see. So to me, this is similar. And then if you want to do something, and then you need to actually do it in the movies, and then put your mining gears, and these are our mining gears, right? And then in the end, you're trying to get hold of those gold pieces. And I think to a lot of you familiar with protein biochemistry, immunology, those specific genes or proteins are what you're after, right? So you got those, you think you can solve the problem. However, the picture is not that rosy. In Nature last year, there's a very pessimistic editorial article published about the lack of confidence for precision oncology. According to a clinical trial study conducted at MD Anderson, if you simply think that the precision medicine is about getting the drug target, sequencing the mutations, and pair it with the right drug, and only 2% of people will benefit from that simple-minded sequencing. And they talk about maybe expanding the population, but I think the diversity of disease is a lot more than what we have imagined. So you can list all of these challenges. The challenges are the disease are very complex. We don't have limited therapy choices. Patient-collected data have lots of noises. And we don't actually have these so-called the real big data that the e-commerce people have. They have continuous monitoring of you day and night, but we only have snapshots. So this is an example. One of my former postdoc did 1 year of visiting with me and did this study and published the result in PLOS One. This is a very practical use of bioinformatics. So you start with a biology problem. So there's a bacteria, infectious, and then you isolate the genes, and then you look at the microRNA expression profile of that infested host, and then you get a whole list of things, and you can do all kinds of annotation, and you publish a paper. But the question is this real bioinformatics that solved the problem, right? I believe the challenge right now is one of the the person in the audience asked earlier. How do you actually bridge the gap between the big data on this side and the wet lab work that you do on this side, right? So we are building some pipelines, and we're very good at doing primary analysis today. These primary analysis allow us to format the data, extract useful information using statistics, and then give you some list of stuff. I think the thing in the middle is something that we're quite weak. It's called a downstream analysis. It involves all of the clustering, network analysis, pathway, gene set enrichment. I'm not going to talk about today, but there will be classes if you're interested, right? Then talk to me later. So I think that this part is really, really messy and really, really difficult, right? To the point that if you're not careful, and then even for informaticians, we can get burned. And there are prior precedences why pipeline has been talked about and then has not been very helpful in drug discovery. Going back 50 years, they talk about pipelines of screening compounds, starting from 10,000 compounds, and then going down sometime 12 years down the pipeline of clinical trials. Eventually, you'll be lucky to get one chemical drugs. And then why do you think that we're so lucky, even in the precision medicine, that we can get a single magic bullet, right? So I'll continue with my analogy. Imagine these are all the wonderful sand or the omics data you have collected. You say, well, I'm done with my NIH study. Now I think the gold is in there. If you're a graduate student, you're going to do this, right? Labor intensive, good luck. All right. If you're a graduate student in bioinformatics, you're going to build something that is really impressive. It's a castle, but who cares? I think that if I map these one-to-one, and then you'll see what I mean, right? So you have a list of stuff that you got from omics. Here you got the markers, and you think the marker is pretty good, but then when you test it in new cohort, new population, it doesn't work anymore. And then here is some really impressive clustering result. You can bend it, but did you really extract the true biology out of it? Well, I think that there's an opportunity, and the opportunity is what I call the glass blowing or the glass making. I think I'm running out of time, but I think that there's a wonderful piece of a YouTube video. If you're interested, I can share with you. Essentially tells the wonderful stories of glass making. How many of you have seen the glass making before? Okay, wow, this is Birmingham. So when I watched that, I was really amazed, because you start with the sand, and then you mold that sand into glass, and then you blow it, and then you rotate it, and it's a wonderful piece of glass coming out. And the one thing that made me think is the profound transformation of the sands. It's something that a true bioinformatician should do, is that we should take the raw data and make some profound transformations so that it can really tell the story. right? So what is so profound about the data? I think it's a systems perspective. And the systems perspective is what systems biology is all about. You're not just looking at a piece of the data, you're looking at the structure, you're looking at the different component, as well as the interrelations between the component, protein-protein interactions, gene modules. You're not just looking at the systems in static view, and then you're looking at how it changes over time. That's the dynamics. You're also looking at what made those changes and the control molecules and microRNAs or the epigenetic controls that bring about all of those changes. In the end, you can actually do synthetic biology and drug design. Okay, so we have a whole series of approaches and using network biology. Essentially, these look like graphs, and the nodes represent genes in the most cases, and then the connections represent their correlations or interactions, however you define it. And we can study how these network changes and then how the network can be controlled so that go back to infer the biological function. This is one example, what I call the gold mining example, right? Remember the previous analogy I told you? So in gold mining, we were able to reduce the data that relates to the drug target that have side effect into this small network. All the side effect, so the drug targets are circled green in this plot. And then whenever there's a PubMed article that talk about this gene may have a cardiotoxicity, then we color it red. Right? Whenever there's interactions, and then we have different lines between them. And then some of the drugs, all of these drugs will have a certain degree of just a cardiotoxicity. So you can see some of the drug, even though they're not designed directly on the cardiotoxic proteins, but just by guilt by association, you know you have to be extremely careful, right? Because these drugs, these proteins, once perturbed, and then they can radiate their impact to the neighbors. So this is what I call the gold mining. You're not transforming the data, but you're annotating it. You're actually sifting, teasing out the noise, and then eventually you see some story. Useful, okay? And then there's another castle building example. So in this case, each of the nodes here is actually not a single gene. It's a group of genes that we tested to be enriched using gene ontology analysis. And then lots of the lines here are actually directed regulations. And then the up and downs are the enriched genes in that gene ontology categories that are significantly up or down regulated using red or green. Okay? So this is impressive castle, and then we actually published a paper and we argue this is for myelodegenerative cells, and then in the cancer immunity, these cells were elicited to become a tumor helper cells, and then we try to understand what's going on. So good insight, but not truly transformative, right? And this is what I call a glass blowing example. A glass blowing example, you can almost forget about informatics. Everyone can look at these pictures. And if I tell you that the 3 pictures are for Alzheimer's disease, the first one is the incipient mild cognitive impairment versus normal cases, the molecular panel, imaging from the molecular panel. The second one is moderate against control. The third one is severe against control. What can you tell about these 3 conditions? You can say, oh, your A1 and A2 peaks are consistently up. Okay, they must be diagnostic or at least just symptomatic of these disease. And then there are some peaks that are actually there in the early stage and then disappeared and replaced by other peaks. And these can be staging markers. And then some peaks are actually rise up in height, and they could be prognostic markers. In fact, we discovered this almost 10 years ago. We were in Indianapolis at Indiana University talking to Eli Lilly, and Lilly were doing a clinical trial at the time. They were saying, oh, these are innovative, interesting, but we're already in the late stage clinical trials. We don't know how to incorporate these and how the FDA would be receptive to introducing a newer technology. So many of you probably know that the Lilly has a failed Alzheimer's disease drug that target amyloid beta. And the interesting story of that drug is that previously they have a failed phase 2 that target this drug. But then in their statistics, they found something really interesting. The people with late stage Alzheimer's seem to benefit somewhat from the drug. So instead of using a molecular, more precise technology like this, they decided to use a survey to figure out, well, which patients are more advanced and which are not. And unfortunately, Lilly didn't succeed at phase 3 clinical trials, and that was more than hundreds of $millions in waste. But we're still actually pursuing this story as Lilly is. But you can see that this story, if you look at underneath the hood, there are gene sets, networks that build this terrain. And this has already gone through lots of transformation. What are the transformation? You don't have to go through the details, but we have different steps of generating these, transforming the raw data from the gene expression files that has a list of differentially expressed genes, and then added the network data, interaction data, layout, and parameters, and finally come out with this. So how do we tell the story? I think the story can be told together between a data scientist and the biologist at the iterative cycle sense-making stage here. The data science, we have the pipeline coming in here. And the biologists actually can use tools to validate the biology. And then we come to an iterative platform such as Gene Terrain, and then we can just exchange ideas, okay? So I think the whole idea is that you look at a figure and the software allow you to not only see the picture, but also the gene details. So it's the topological information that matters and expression information that matters, okay? How many more minutes do I have?
As many as you need.
Wow, I got the special treat.
That's Lowe's, yeah.
Okay.
I told Sunshine to have you keep going.
Okay, all right.
As long as the panel discussion ends in 25 minutes.
Okay, all right.
Yeah, you're pretty much on time. It's
Okay.
2:04.
All right.
4 minutes over.
Okay, so I think I won't go through the detail, but I think there's lots of the informatics behind the creation of these tools to make sure that we can see the group-wide changes after tuning the parameters. And sometimes the group-wide changes can be more profound than the single changes. So for a tool like this, if you can adjust the parameter, you can not only do the single gene analysis, but you can also do this gene set-based analysis. But how do you get the gene set together? Well, also underneath the hood, we have these complicated layout algorithm that controls, in this particular case, if you look at this area, how well the space can be filled versus if you're controlling the parameter and more toward this side, is how the hub-centered community can be highlighted, right? So there's a balance that has to be struck, and then we make many algorithm decisions behind the scenes. And I'm still not showing you any formula today, so I'm pretty proud of myself. And then in addition to that, the gene terrain can incorporate prior knowledge of gene set annotation. If you're using the standard layout, and all of these colors are the different prior annotation gene set, you can see that they can be spread all over the places. For example, these may be immunity, these may be the cancer-related, cell cycle-related. But then, I mean, that layout is okay, but not ideal. But if you do a similarization to optimize, and you can see that within this subtle fine-tuned network layout, and the gene set that are more or less belonging to each other are grouped. And these actually provide the basis for seeing more biological, meaningful picture at the end. So we hide a lot of complexity. And let me show you quickly 2 case studies. One is that you can really use these at the very, very high level of just using it as a picture, just like an X-ray or ultrasound. And in this case, we did a study for a company, and they have non-alcoholic fatty acid samples, and they have normal samples, 32 of them total. We created this terrain image digital knowledge map. And you can pretty much see that some of these dots actually seem to be diagonal. Some of them are just horizontal. These are exactly indicative of the sample type, whether they're non-alcoholic fatty acid syndrome, fatty liver syndrome, or the normal liver sample. And these are some of the early diseases that are difficult to detect non-invasively. And then on this map, we can show clear distance. In fact, if you were to do one sample learning, and then this is almost like a trivial case, 100% sensitivity and specificity. Okay, let's go to another example. That's the last one, I promise. There's a paper published in Nature. So one of my students in biology came up to me and said, let me do a rotation project. I said, well, find me a challenging paper, science paper with a lot of data. So he found this. This is an aggregated schizophrenia GWAS study that contains 36,000 cases and 111,000 controls that spans several GWAS studies. And it's one of the largest data set available today. If you're not using the type of glass-blowing visual storytelling techniques, you have to rely on statistics. And the standard statistics for these is that you have to set the threshold to be really, really high so that only the significant SNPs show up. In this case, not so many show up, right? 108 loci show up. But we started with a mission, let's be more permissive about the loci, and let's try to put the story, essentially get more sand, and then let's make more glasses. And that's our pipeline for generating it. We start with almost 10 million raw data, and then filter it down to 60,000 SNPs, down to 2,000 genes, and then got the filtered genes and the SNPs, calculated the SNP contribution score around each gene, and then we build a terrain like this. So what do we use? What do we do with this terrain? We look at the original paper, and we use that gene terrain, or we look at, of those that are highly ranked, how much knowledge can we actually is overlap with the original publication. So you can see these drug targets are ranked very high from our gene terrain, and then we were able to reconstruct the drug target. And then lots of the other, the red categories are also found with a lot of additional information that we can supply to a person like a biologist in the end. So after a very, very long story, and I think my message is, well, maybe we should all learn glass blowing today. But actually, my real message is that let's think about how to use data wisely so that we can tell more stories with more sophisticated bioinformatics. And with that, I want to thank all the members of my lab, especially Kevin, Zongliang, and Zhenyu that contributed a lot of the unpublished data that made to the slides today. Thank you very much. Yeah, let's do a panel.
Okay, so he prefers to start the panel. So why don't we have all the speakers come at the table, and then everybody can ask, not just Jake, but everybody, all the speakers. Okay, everybody, all the speakers to that table, and we're gonna say, oh, so our moderator of our panel is David Crossman. He runs the Heflin Center Bioinformatics. So if you have a question for him, and you can ask him too. So David, okay, so all the speakers there, and you guys can ask anyone.
Okay, hold on here.
Robin was going to go first.
Yes, I did say Robin.
I even promised Robin that she would go
Hang on,
first.
just hang on. No, actually, I wanted you guys up behind, yeah, because if you get in front of this, nobody's gonna be able to see you. I guess, yeah, back here. And then just use this. Whoever it is. Whoever's in, it's already on, ready to go. Hang on just a second.
Who got the microphone?
Robin.
Hang on. Okay. Okay, this is for the woman who's talking about the single cell. I'm sorry, I don't have your name. If you, Mint. Okay. So the first technologies you were talking about were primarily Fluidigm, right? And then the second one looks sort of like a mechanical grab the cell kind of thing. Is that correct?
It's done by manual.
Right, it's manual. So you have to actually pick up a cell. So it's done microscope aided somehow. And can you use fixed tissues for any of your technologies for RNA-seq?
This uses the pipette.
Okay.
Uses a special pipette to just use microscope and the pipette to pick the single cells.
Right, and so can you then use fixed tissue?
Maybe not work for T cell. It's used for cell lines.
Ah, okay, thanks.
Okay. This is for all of you, whoever wants to take it. Bioinformatics, system biology, pathway analysis, all of that under the same umbrella. What are the measures of rigor and reproducibility that you apply to your work? I'm a statistical geneticist. And 10, 15 years ago, the top echelon journal said, if you do not have power, you do not have large enough sample size, if you do not correct for multiple testing, and if you do not have an independent replication built in your study, we're not sending your paper out for review, period. So I want to know, and that has been ingrained in my head. That's how we work in our lab. I want to be convinced that you have measures of rigor set in your model, so I can come with you with all my GWAS or EQTL results, and then you can come back with a solid story that brings everything together.
I will take it first. I think it's a very important issue. I, in fact, think that the major difference between a bioinformaticians and a computer scientist is that bioinformaticians tend to think statistics along with the algorithm. And computer scientists usually find a more efficient way of doing things, but then tend to be a little weak. For example, every step along the way when we're building a systems biology model, we come up with some kind of a list, and we always use a statistical model to guide us. Where do we train the data before we actually go on to the next step? What is the characteristic of the noise? And then I think it's very important. Sometimes our models are not based on very strong theoretical underpinnings. They're very pragmatic. And this is where the statisticians will be very helpful to bioinformaticians. And I look forward to my colleagues Xiangqin Chen to describe some of that in the second session.
I'd like to add to that, that there's, from the point of view from working on different pipelines, that there's a lot of quality control measures you can take along the way before you get to your derived data. And it's very important not to treat everything like a black box and have methods where you can validate and verify what you're getting at each step of the way. The second point is with data visualization, which is really crucial for a lot of this kind of complicated data. And it's a great way to convey, as Jake just showed, to condense really complicated information. It's also important to keep in mind that in a lot of traditional kinds of visualizations, it's shown without context. And so when you look at a heat map, you don't really always have a great reference to understand how dramatic an effect something is. And so I think you have to be very careful about how you interpret that kind of information and maybe perhaps try to understand, maybe using random data or resample data, how robust that kind of response is. So I think when you're dealing with visualization, you should keep in mind those kinds of factors as well.
I just want to go on the record that you did not convince me. You did not answer my question. And the fact is that I think we're here so that we can collaborate, right? So you mentioned mathematicians, statisticians, and then the disease people. I am the one who goes out and gets thousands and thousands of people with the disease. So when you are showing me a GWAS that has come up with more than 100 hits, that's not a single GWAS. That's like tens of GWASs together. If you come up with a pathway out of that, I would like to know that that pathway is solid. Unless you convince me, I'm not buying it. I'll give you one other example. A new way of doing system biology and pathway analysis. Just about every disease that it got applied to came up with immune system. What does that mean? That just means that immune system is affected in disease. So I am leaving you with, I totally believe what you do is crucial, but as a friendly person, I'm telling you that down the line, you're going to have to face people like me. We're going to say, show me the money, show me that it replicates.
I have three very practical questions. So the first question, I thought it was for Alex, but now I realize probably it was for the others and also for the institute. So Alex, you showed two very beautiful samples about the B-cell repertoire sequencing and also the multi-parameter flow cytometry. But at the same time, on this campus, looks like there's an increasing need, particularly for the immunologists, for the integration of the data sets of ChIP-seq, RNA-seq, and also the ATAC-seq. So my question is specifically about the ATAC-seq, because on the way with Amy to Canada last week, I realized that no immunology lab right now on this campus has the capability to analyze the ATAC-seq data to kind of for the real new discovery. So my question is, what capability do we have on this campus right now for the Bioinformatics Institute to analyze the ATAC-seq data for the new discovery? So that's my first question.
I know several people on campus that analyze ATAC-seq, and several investigators are in this lab that do ATAC-seq.
No, we are doing in lab as well. We do single gene by gene, not by systematically to look for the new discovery. I think we haven't reached that level. It's like even Amy's lab, right?
Right.
No, we don't reach that level. We just look for, we use it just as a map. Right. So that's my first question.
Well, I think there's the capabilities for dealing with the primary ATAC-seq analysis. I'm not overly familiar, being fairly new, is distributed, that I think the labs often do it within their own labs and put something together. As far as the other point about integrating data sets, I don't know right now of a centralized place where that happens.
Yeah, so I mean, so that's my second question. Again, so that's why I realized the question is not only for you, right? So Jake mentioned that we have the primary analysis, looks like the chip-seq data and RNA-seq data. We have the pipeline, right? If we do the in-lab analysis, how many of those tools are free for us? Because in the bioinformatics analysis for each lab, the headache is paid by hour, right? And that's endless. It's just like the question there. Because the question answer to you is everything Python eventually needs to be validated.
Exactly.
Right? No. So I understand that. But for each individual lab, there's if it's fee by service, it's endless. It's just too expensive. So if we do the in-lab analysis, how many of those tools for the primary analysis will be free to us?
Yeah, so that's a good question. A lot of the free tools are out there. So Galaxy is one of the prime example. It has a limitation, but it can take care of a lot of your tasks quite well. And if you actually are willing to pay for some of the low-cost tools, like 7Bridges, and they have their tools and that are very streamlined. I think last month CCTS has invited a speaker from Utah, and they also have their tools. I think the free tools have limitations. You have to know how to use program to stitch the result together. But I think in the second panel discussion session, Elliot is going to moderate that. I think Elliot's group and I at the Informatics Institute have been talking, how do we make some standard reference implementations and push our experts pre-built pipelines as a reference template onto Galaxy so that everyone can just do self-service. You don't have to come to us unless you have a plumbing problem or the really disaster problem, then you can come to us. Right? That's in the plan.
So there are a lot of questions in here in what you've been asking. I'm just going to answer one question right now. None of it's free. And I think there is a general misconception overall that analysis should just sort of come with a program, that it should just be provided by the institution. It should be provided by bioinformaticians throughout the world overall, because you need it to analyze the data. And that's really the wrong way to think about it. Bioinformatics, the ability to take raw data and analyze it, needs to be an integral part of the planning of any particular experiment from the beginning. And that planning starts with, how do you fund what you're doing? So when you write your grants, when you get your pilot studies, the analysis of the data has to be thought about, has to be included in how you budget for everything else, including personnel, including pipette tips, including enzymes. Bioinformatics needs to be part of that.
Let me add a comment there. So,
my ex-chair used to say, that you either bribe us or seduce us. So this is one way of allocating budget for this. The other way to actually find a bioinformatician and make him excited about your project. So you'll be doing this work for free. And we'll write a grant with you, actually consider him as a peer, as a colleague for the inception of the project to the execution of the project till the end. So it's not your project, it becomes his project too.
I want to add to that. The third option, actually. Even though we do the disease end, we also do all the analysis in our lab, because you can see from my gray hair, I've been in the business for a long time. I always feel like something gets lost in the translation between the people who are actually doing the disease and people who are doing the analysis. So I feel best that people who are in my lab understand the analysis and they can conduct it in the lab so that they know exactly what's going into it and what's coming out of it. So as a third option, I would like to propose that you have tutorials where we can send our graduate students to you to learn how to do what they need to do.
So there are some of
We will
those.
pay for it. We will pay for it.
I mean, there are some of those courses out there that, and then I know Jake is planning on offering more of those. So they are becoming more available.
I'm not asking for courses.
Well, not even courses,
They're
but
coming in with a project
right.
and being trained as to how to
right.
Yeah, no, I don't consider that the third option. I consider that the first option. I think that's the only way that computational analysis, that the ability to analyze modern day and future data sets is going to be done. Every individual, every graduate student, postdoc, and faculty member has to understand the computational requirements of analyzing their data sets and producing results. And so the courses are absolutely necessary, but the courses are not only for the people who want to get advanced training in bioinformaticians. We have to make a requirement, as we do biostatistics for every graduate student. I think we have to make bioinformatics a requirement for every graduate student now. Otherwise, they're just going to be, treading water and not producing the rigor necessary to really ensure that the data they're producing answers the question that they started with.
So I have a question on indel analysis. There are different kinds of genomic rearrangements, some like transposable elements and repeats and all sorts of fragile spots in the genome. Do you treat them differently? Because it might increase the power of indel analysis tremendously.
Yes, yeah. That's a good question, because the nature of our genome is quite complex. There's low copy repeat regions. There's all kinds of rearrangements events happen in the different type of regions. So yeah, I will treat differently, and for different problem, I will try to solve it differently. Yeah, that's what we are doing. No, maybe we can just focus on the what type of variants you are interested in, and we are working on this specific type of variants, short variation or rearrangements. Different type of rearrangements have different signatures. So yeah, I will first treat different types of variants first, and then focus on different type of regions. So there are different layers of treating or dealing with the question.
Oh, we've got a couple more minutes.
Yeah. So Elliot, I think you misunderstood me. I'm never asking for free, right? I'm just asking some basic tools, and I totally agree. I totally agree with this training part. Okay. So my last question is a very practical question is for me. So you give two samples, 67 cells and 509 cells. What's the bulk part of the cost for the single cell sequencing for those two projects?
For the cost?
Cost, yes.
Yes. For the case one, use the FluDAM C1 system. We just for one use 196 96 well chip from the cell suspension to the end the data analyze. It maybe cost like more than 6,000. But for the second case, it's only 2 or 3 single cells. It's less than $200. And the third, for the ICL8 140 ICL8 system, it cost about 6,000 for 1,000 single cells.
So I guess I have a question about the visualization tools and basically trying how to best represent your data, because I think that's a pretty large problem that a lot of people face, especially if you're like me and you are not as familiar with trying to represent data in a meaningful way. And so I guess I've heard mixed feelings about some of those representations, about all these complex systems, biology maps, and it's all beautiful, but at a certain point, how interpretable is that? And do you expect that this is going to be a trend where more people are going to start moving towards this direction? And yeah, I guess I'm more of a all the beautiful colors, but how meaningful is that for a biologist who is looking at this and trying to extract important information so we can go back and further continue the, I guess, wet bench aspect of things?
Well, I think for the visualization, there are actually different type of visualization. There are visual analytics whose primary vision is to enable leading discovery for the expert. There are actually visualization that are meant for non-expert general audience to also be able to appreciate what the data has. So these are completely two different directions. I can only talk about the gene terrain. When we started gene terrain, our goal was to actually use this visual analytics capability to do something that has not been done before. So our mission was never to make it easy. Our mission was like the analogy that I was using, glass blowing. We were trying to create new art and science as long as we can be validated. But I think at the same time, a company has been spin off, which I didn't mention, and they are actually taking a different route. They're making it online, they're making it easy, and making most of the functionality for free. It's geneterrain.com, I guess, but if you're interested, I can share with you the link, and then they are coming up with versions.
I want to comment, and yeah, actually, there's a different type of data, and I'm focusing on genomic study or epigenomic study. So there's some figures or plots we can generate, like to show the general or overall. So yeah, you can find some patterns based on these type of figures. And the other things that we will also focusing on the specific, like the differential expression genes. We'll give you the list that are differentially expressed. You can select those top differentially expressed genes to validate or to do further validation or subsequent experimental analysis. So there's a collaborative and there's some interactive way that we can interpret the data and try to also make beautiful figures. I think figures are useful and for publication purpose or overall pattern or whatever. Yeah, from genomic or epigenomic part of view.
All right, well, let's take our 10-minute break here. I think actually we were in that 10-minute break, so let's be back.
Sorry. I'm trying to start that as well.